The overall goal of using image analysis software in developmental biology is to obtain quantitative data for mechanistic models of biological processes.
This method can help answer key questions in developmental biology, such as how can mutant phenotypes be quantitatively analyzed.
The main advantage of this methodology is that it provides researchers with the ability to identify alterations in biological processes and developmental phenomena in an unbiased way.
Though this method can be used to provide insight into development and morphogenesis of C. elegans, it can also be applied to other model organisms such as Drosophila.
Demonstrating the reconstruction of cell shapes with iMod will be Christina Lehmann, a PhD student in my laboratory.
After installing the iMod program, open the Unix shell and type in iMod. In the iMod window, select the file with the appropriate raw data from which to generate a 3D model. And use the information window to locate the bottom and top of the objects in the Zap window. In the information window again, trace the cell outlines, creating the first contour at the top plane of each cell and taking care to create a new object for each cell. Then scroll down in Z and create a new contour for each Z-plane. And subsequently create a new object for each cell.